Okay, good afternoon. I think we should start. So the first afternoon speaker is Elisa Kallioniemi. Elisa is at present assistant professor at the Department of Biomedical Engineer in New Jersey Institute of Technology. Elisa has a PhD in physical medicine. Then she did a postdoc also working on the field of TMS to treat patients mainly, if I'm correct, developing protocols to evaluate cortical plasticity and therefore how you can use cortical plasticity to induce improvement in patients. Today, Elisa will talk about the use of TMS-EEG in the aging, the lifespan. Sorry. Okay, please welcome Elisa to.
Okay, so hi everyone and good afternoon, and thank you so much, Carlo, for the kind introduction and for the invitation to this amazing workshop. Is this better? And thank you, of course, organizing this workshop. I mean, I've learned a lot. So my name is Elisa Kallioniemi, as Carlo mentioned, and I'm from New Jersey Institute of Technology. I have no conflicts of interest to report, but I must disclose I have a very bad cough. So if I start coughing unexpectedly, please ignore me. I will do my best that my voice holds during this presentation. In this talk, I will present the overview from our recent review, in which we looked at how TMS-EEG changes, TMS-EEG responses change across the lifespan, and how these responses are impacted by abnormal brain maturation and aging. So first, just a little bit about the importance of applying TMS-EEG to study normal and abnormal brain maturation and aging. So currently, there is an increasing prevalence of neurodevelopmental disorders, especially, for example, autism spectrum disorder and attention deficit hyperactivity disorder. The reasons for the increasing prevalence are quite contradictory, so we don't really know. So there are some explanations, for example, changes in diagnostic criteria, also how we report these findings, also changes in culture, environment, and socioeconomical status. Regardless what is the reason, this is a problem. Then at the same time, we have an increasing society. So due to increasing longevity and improved healthcare in the elderly. So this has led to an increased number of chronic diseases in the elderly and disability. So basically we need more understanding and more research to kind of figure out what causes, like what happens in typical or like healthy aging, healthy brain maturation, healthy aging versus abnormal brain maturation and aging. So, for example, we could develop biomarkers to predict if somebody will develop a disorder later on, and potentially create an intervention to kind of like prevent the progression of the disease before it even gets to a severe stage. And then, so TMS-EEG is relatively inexpensive, and it can measure the state and dynamics of the excitatory and inhibitory systems separately. It can also differentiate two different inhibitory systems, the GABA-A and the GABA-B activities, that are often actually associated with abnormal brain maturation and aging, as I will show you shortly. So TMS-EEG is a potential method to study this. So then let's look at what happens in the central nervous system during the first 2 decades of life. So the data that I'm showing here in the figures it shows different structural measures from MRI, and the age range in these figures is from 4 years up to 22 years in the x-axis. The blue line indicates the males and the red line females, and the arrows in the figure show the peak age when this particular brain structure kind of like reaches its peak. So as we can see, if we look at the figure on the upper right corner that shows white matter changes, we can see that the volume of white matter increases linearly with age. It increases slightly less in females than in males. It is not shown here, but there's no differences between the various lobes, so similar changes happen in all our lobes. The gray matter changes, on the contrary, are non-linear. They're very region-specific, and the peak age varies somewhat between various lobes. So here's data on the frontal, parietal, and temporal lobes. They have kind of like inverted U-shaped curves. Whereas on the occipital cortex, here on the lower right, females and males have a different behavior. So with males, it's a U-shaped curve, and for females, an inverted U-shaped curve. Regardless, in both females and males, during these 22 years, the gray matter does not reach its peak yet. And then during these structure changes, there are also substantial functional reorganization, especially in the excitatory and inhibitory systems. Also, there's a reduction in the excitation-inhibition ratio in the brain. So this means that basically our inhibitory system starts to inhibit more spontaneous brain activity in favor of stimulus-evoked brain activity. So basically there's an increase in signal-to-noise ratio. So how these changes, these structural and functional changes can then be seen with, like during brain maturation, can be seen with TMS. So if we look at the most common TMS measure, the motor threshold, which is common almost basically in all TMS measurements or whatever we do with TMS, and it reflects the cortical excitability on the motor cortex. We can see that brain maturation decreases at the motor threshold. So, and this data is with a biphasic stimulation waveform. So here's data from children, from left to right, children, adolescents, and adults. And we can see that the motor threshold in children is about 60% of the maximum stimulator output, and it decreases to about 45 in adults, which is, of course, a substantial change. And this decrease in motor threshold has nothing to do with scalp to cortex distance change, as in fact, as you can see here below, the scalp to cortex distance actually increases with maturation, which would actually mean that the motor threshold should increase, not decrease. So this means that in all studies with children, we usually have to use a higher stimulation intensity if we want to normalize the stimulation intensity to the motor threshold. Considering TMS-EEG, this would of course mean that, for example, that the TMS click sound is louder, which hypothetically would create more stronger artifacts to the data compared to adults. So something to consider. And another thing that happens with brain maturation is that both the interhemispheric and intrahemispheric provocations increase. So here we're talking about propagation of TMS evoked EEG activity, mainly like if we get the response, how much does the area under the curve of the response kind of transfer between different areas. So then let's look at how the TMS-EEG responses change during brain maturation on the primary motor cortex. So here are some responses presented as a global mean field power, which is just basically standard deviation across all the channels. And below the figure, there are some gray boxes that show the statistical comparisons between these three groups. So when there's a gray box, it means that there was a difference between the groups. So as you can see, on the primary motor cortex, brain maturation decreases the responses. So we have first children, adolescents, and then adults if we go from top to down. And mainly this decrease occurs in the response that we commonly call the N100 response. And of course, please pay attention to the scale. So we're talking about tens of microvolts, which is very untypical for TMS-EEG, but the children's response is really enormous. So another phenomenon that is impacted by brain maturation is the phase of the oscillatory response, which can be measured with intertrial coherence, ITC. So this is a measure of how consistent is the oscillatory phase across trials. And here I'm showing ITC on the alpha and beta bands that are impacted by maturation. So alpha is on top and then beta is down. And on the left, we have ITC in children, adolescents, and adults, and at two different time scales, so from 20 to 200 milliseconds and from 200 to 500 milliseconds. And on the right, we have the differences between these groups. And the light dots, they represent the skull positions where the groups differed. And everywhere where there's a light dot, that basically means that in children and adolescents, the ITC was higher than in adults, and there were no differences between children and adolescents. So all this between children and adolescents and adults, always the adults had lower ITC. So basically the phase goes more inconsistent as the brain maturates. And we can see this phenomenon in both of the the alpha and beta bands, but also in both time spans. So it's from 20 milliseconds up to 500 milliseconds. So one question, of course, comes to mind when we're looking at these results is that would there be an association with the changes that occur in brain structure? As I mentioned earlier, there are huge changes in brain structure, and we looked at this earlier. So here are some findings from a study where we looked at TMS-EEG responses and MRI features. So these are all MRI features from the stimulated hand knob. And we looked in TMS-EEG, we looked at both the global mean field power, so data from all the channels, which is here in blue, and then local mean field power, which is only the data from the hand knob, which is shown here in right. And the data is not here separated to groups because we just wanted to see, is there a connection? And what we found was that, that with both the local and global mean field powers, they correlated positively with some of the white matter features. So we found positive correlations with mean and diffusivity, MD, which is basically a measure that how diffusivity, the diffusivity, regardless of direction. We also found correlation with axial diffusivity, which is diffusivity parallel to the axons. So the higher these diffusivities were, the higher the local and global mean field powers. Surprisingly, we did not see any connections with other measures, for example, the cortical thickness. So gray matter did not have any correlations, only the white matter. So in addition to merely looking at these local features on the hand knob, we also looked at how the whole brain volumes impacted this, like was there a connection to the whole brain volume? And we looked at both the absolute and relative brain volumes in both white and gray matter and CSF. So here on the left, we have relative white matter volume, so white matter volume that has been normalized to the whole head. And we can see that both the local and global mean field powers correlated negatively with relative white matter. We also found that the global mean field power correlated positively with the absolute gray matter volume here in the middle, and that the local mean field power correlated negatively with absolute white matter. So basically, the main message that I want to convey to you is that at least during brain maturation, there seems to be a connection between TMS-EEG responses and the changes that we see mainly in white matter, but of course some small changes in gray matter as well. So structure matters. So now that we have looked at how TMS-EEG changes are impacted on the primary motor cortex, let's see what happens on the more frontal areas. So here's data from where TMS was targeted to the right superior frontal cortex. And in this data, we did not normalize the stimulation intensity to the motor threshold, but we made sure that the estimated electric field was the same in all groups. So this is slightly different compared to the rest, to the previous study. And again, the responses are shown as global mean field power. And so in this, in the superior frontal cortex, we also saw that the responses decrease from when you go from childhood to adulthood, but the changes are very smaller. So if you look at the scale here, we're talking about a single digit, whereas on the primary motor cortex, we were looking at tens of microvolts. So slightly different phenomenon. And on the right superior frontal cortex, we did not see any differences or impacts to the face. So intertrial coherence was not impacted. Right. So that's basically currently all that we know as the field in what happens in healthy aging, which is of course not a lot. So I mean, there's still a lot to be done. So let's look at how are these responses impacted in children with neurodevelopmental disorders. And again, also in neurodevelopmental disorders, we're talking about a handful of studies. There's not much. So let's first look at the attention deficit hyperactivity disorder. So in children with attention deficit hyperactivity disorder, the N100 response may represent a promising biomarker. So here I'm showing data from Brockman et al., a study from 2012, in which they studied ADHD and control subjects between the ages of 8 to 14 years old. They did the study during rest and during a kind of like a task, movement preparation task. And the black curves show the responses during rest, and the red curves during this response preparation task. So the main finding from this study was that the N100 amplitude was smaller and its latency was shorter in children with ADHD than in controls. Also, only in controls, the N100 response was significantly reduced by this response preparation task. So there's a small difference here, but it wasn't statistically significant. And similar findings have been found in other studies. So far, there's in autism spectrum disorder, there's only one study by Jarzak et al., in which the authors looked at interhemispheric signal propagation with TMS-EEG between ASD and healthy controls. And the hypothesis of the study was based on, because there are some previous MRI results showing that there's some abnormalities in the corpus callosum in ASD. But surprisingly, the study did not find any differences between autism and healthy controls. So TMS-EEG did not show these structural changes in the corpus callosum. However, in both groups, the interhemispheric signal propagation increased with age. So basically, in this case, of course, TMS-EEG may not show correlation with structure. There is also one study that evaluated TMS-EEG responses in pending childhood epilepsy with centrotemporal spikes. And these individuals have rare seizures that usually get initiated from the primary motor cortex. They also may have language deficits. And in this study, the authors evaluated rTMS-induced plasticity and excitability in these individuals. Basically, the rTMS was given with 1 Hz, so the so-called inhibitory protocol. And what the authors found was that rTMS induced an increase in N100 amplitude in those individuals who had a larger baseline N100 amplitude. And those who had a smaller N100 amplitude at the baseline, there was a decrease caused by rTMS, shown here on the left. And these effects, so how the individuals responded to rTMS also correlated with language deficits. So here on the right, so basically, if you had a decreased N100 amplitude at baseline, you had worse language learning abilities, whereas if you had an increased N100 amplitude at baseline, you had better language learning abilities. Of course, this is purely a small study, so still a pilot finding. So that is actually all what we know about healthy and abnormal brain maturation. And as you can see, it's not much. So I mean, for all the young researchers, there's still a lot to be done in this field. So taken together, so the results that we currently have demonstrate that TMS-EEG can be applied to pediatric populations and may be feasible even in children under school age. And TMS-EEG indices change from childhood to adulthood, suggesting that TMS-EEG could attract healthy and abnormal brain maturation. And of course, notably, none of the reviewed studies reported any significant adverse effects. And most published developmental TMS-EEG studies so far have focused on the N100 component from the primary motor cortex. And they have found that it could be a promising biomarker. And although, like as a TMS-EEG field, we always think that N100 amplitude could be an inhibitory response, mainly related to GABA-B. And now recently, of course, like there has been some evidence that it could also be impacted by artifacts. However, I just want to point out that all the studies were done in adults, so we don't know whether the N100 response in children is the same as the N100 response in adults. So that is also a question mark. And overall, all the studies that we have currently are cross-sectional. So we have there's groups that are separated by age, and none of the studies have looked at differences between females and males. So basically, we still need more longitudinal studies. For example, it would be amazing to have a study where these individuals would be followed several years when they go through a brain maturation. Right, so that was the brain maturation. So then let's look at what happens in healthy aging. So it has been widely found that the volume of the brain and its weight decline at the rate of around 5% after the age of 40. With the actual rate of decline possibly increasing after the age of 70. And here are some examples from the lateral prefrontal cortex, primary visual cortex, and hippocampus. There are also some changes, for example, in dendritic arbor, so dendritic structure, and synapses. For example, there's a loss of synaptic plasticity when we age. And substantial functional organizations in our brain. So as an example, I'm showing activations in young adults, low-performing adults, and high-performing older adults during a neural encoding task. So as you can see, the low-performing older adults exhibit a similar pattern as to young adults. So the two figures from well, this one doesn't really work. So the two figures from starting from right, whereas with high-functioning older adults, the activations are very bilateral. So basically, there's a huge difference, and this is a phenomenon that can be seen in many different tasks. Everything becomes bilateral. And of course, we all know that aging is associated with cognitive decline, and declines are evident in all domains, with the exception of preserved verbal and numeric ability. However, despite all these kind of like negative things, we all know that the brain is able to adapt and change and learn new skills throughout the life. And even recover functions after nervous system injury. And just to clarify what I mean by healthy and pathological aging, so in this presentation, basically we're looking at those who remain healthy, so the green line here in the figure, versus those that either develop mild cognitive impairment or Alzheimer's disease. Okay, so the main finding from all studies so far evaluating healthy aging are stating that healthy aging does not really impact the TMS-EEG response morphology. So basically, as we age, we still have all the typical P30, N45, and so on and so forth responses, so there's no changes there. However, aging impacts the amplitude, timing, and topography of these peaks. Also, experimental paradigms and tasks differently modulate the peaks in younger and older adults. So here on the left, there's typical responses from the primary motor cortex in young and elderly. So the young are shown in blue and the elderly in red. And then here on the right, there are the differences between these. So this is a study from a study Ferrari and co-workers. So if we look at, and Here on the right, so first is the young response, elderly response, and the third column is the differences. And when there's a white cross, that's a statistical difference between these. So first we can see that there was substantial differences in the P30 amplitude. So the P30 amplitude was larger in younger adults, close to the stimulation, but on the contrary, it was larger in older adults if we go further frontally. The N45 component was smaller in the elderly, mainly in the central and parietal regions. The N100, on the contrary, was larger frontally and smaller centrally in the younger adults. The P180 response was smaller in adults, in elderly adults compared to young. And the N280 was larger at the stimulation site in the younger group and larger frontally in the elderly. So basically, the authors concluded that normal aging may lead to changes in excitability of specific neural circuits, and that, for example, the increased excitability seen in the prefrontal cortex at 30 milliseconds, so the P30, they suggested that it would be compensatory mechanisms for the decrease in excitability in the primary motor cortex. And however, some of these findings were contrasted in another primary motor cortex study. So here I'm showing results from Opie et al., a study 2018. So in that study, the authors recorded TMS-EEG responses from left primary motor cortex in younger and older adults with suprathreshold stimulation. So in the single pulse study, the P30 was unaffected by age, so it was opposite that was found in the previous study. Also, the N45 was increased in older adults. The previous study showed it differently. And then the N100 and P180 showed altered spatial distribution. The authors did find, though, that the P30 response had a shorter latency, and the P180 had a longer latency. In addition to the single pulse stimulation, the study evaluated long interval cortical inhibition, LICI. And this is a paired pulse paradigm that refers to suppression of activity. So we get two pulses, and the response to the second pulse is typically smaller. And we think that it reflects GABA-B-ergic activity. And in this study, the authors used LICI with two interstimulus intervals, so 100 milliseconds and 150 milliseconds. And what the authors found was that inhibition of N100 and P180 was increased in older adults following both of these protocols. And the last study in normal aging that I would like to show you today is from Noda et al. 2017, and they evaluated short latency afferent inhibition and how it's impacted by aging. And for those of you who don't know what short latency afferent inhibition is, so it's a protocol in which we combine TMS with peripheral electrical stimulation, and it is thought that this protocol reflects cholinergic activity in the brain. And again, we can see the responses as a modulation of the second response. So in this study, the authors measured short latency afferent inhibition from the primary motor cortex, shown here on the right, and also from the dorsolateral prefrontal cortex, shown here on the left. And I've basically put a red rectangular on the main findings. And also the age groups were, so in the younger adults, the authors restricted it was from 18 to 59 years old, and older adults were above 60 years old. So, so these figures show the group differences between younger and older adults in the modulations. And what the study found was that older participants showed significantly less modulation of the N100 component in both the primary motor cortex and dorsolateral prefrontal cortex compared with younger participants. In the primary motor cortex, also the N45 showed less modulation in older adults compared to adults, younger ones. And if we look at these TMS-EEG response and modulations more closely and on a continuous age scale, we can actually see that all the modulations correlate negatively with age. And what is really interesting is that the short latency afferent inhibition modulations on the dorsolateral prefrontal cortex correlate negatively with cognitive functions. So here I'm showing data from the same study. So the y-axis shows different cognitive test results. And as you can see, in all of them, there's a strong and negative correlation. So basically, the cholinergic neurotransmitter system has been hypothesized previously to have something to do with, for example, in the cognitive decline of Alzheimer's disease. So if these results would be repeated in another study, it would be, they would still be true. It would be a potential biomarker for cognitive decline. Right, so that's basically all that we know about healthy brain aging. So let's look at what happens in abnormal brain aging. And here I'm showing data from Julkunen et al. 2008 study. So in this study, the authors targeted TMS to the primary motor cortex with suprathreshold stimulation in subjects with Alzheimer's disease, impaired the mild cognitive impairment, and healthy controls. And the healthy controls were age-matched. And what the authors found was that the global mean field power was reduced in Alzheimer's disease compared with the controls during the time span of about 30 to 90 milliseconds after the TMS pulse, and also 200 to 80 to 400 milliseconds after the TMS pulse. And they also found a significantly reduced TMS-EEG, the P30 response in Alzheimer's subjects, whereas a subject with mild cognitive impairment showed increased activity in the P30 response. And in a subsequent study by the same group in which the data was further analyzed, the authors found significant correlations between the primary motor cortex P30 amplitude and dementia rating scales in both individuals with mild cognitive impairment and Alzheimer's, shown here on the left. Also, another study by Bagattini et al. In 2019 had found similar things, but on the dorsolateral prefrontal cortex TMS-EEG. So there as well, the higher P30 amplitude predicted poorer cognitive and memory performance. What I'm not showing is here, but so both of these studies also looked at specificity and sensitivity in identifying these individuals, and both of them showed, so the study by Julkunen et al. Showed that P30 showed good specificity and sensitivity in identifying healthy controls from mild cognitive impairment individuals and Alzheimer's disease, whereas the study by Bagattini et al. showed good sensitivity and specificity of the P30 amplitude in discriminating between mild and moderate Alzheimer's patients. However, contrary to these results, another TMS-EEG study of the primary motor cortex demonstrated that instead of decreased P30 amplitude, there's actually hyperexcitability along the P30 to P60 latency window in Alzheimer's disease. And this is a study by Ferrari et al. 2016. And in this study, the global mean field power revealed an increase in the response amplitude between 24 milliseconds to 90 milliseconds after TMS pulse in Alzheimer's disease. And more specifically, like I said, the P30 and P60 wave amplitudes were increased. What is quite interesting in this study is that the results also show that individuals with Alzheimer's would have an adjunctive peak around 80 milliseconds. It's not very well visible here, but there is a small peak. So this is a peak that does not exist in healthy controls or in any other individuals that we have studied. Of course, like so basically, there's a lot of findings in Alzheimer's disease and mild cognitive impairments, and a lot of these results are contradictory. The reasons could be that, for example, some studies have used newly diagnosed individuals with Alzheimer's. Some studies have used those who are on medication and things like that. So we have promising results, but at the same time, we have very contradictory results. So again, we need more research in this field to confirm which one is, like what kind of findings are more accurate. So before wrapping up my presentation, I just want to show one last study, which I find quite exciting. And I think that as a TMS-EEG field, we should start doing more of these kind of studies. So this is the recent study by Ferrari et al. From 2021. So in this study, basically Ferrari et al. Followed individuals with mild cognitive impairment for 6 years and looked at who will convert to Alzheimer's disease. And they tried to see whether at baseline you could already see changes that could predict which one of these individuals will convert. And during this 6 years, so they had on total 13 subjects, and 7 of these individuals converted to Alzheimer's. And the authors did a lot of various analysis, so it's very hard to see the results here because there's so many parameters. But basically, they found that a parameter called stability of the dipolar activity, which measures the variations of the dipolar activity in the global mean field power over time, was a good predictor of who will convert and who will not. Also, they showed that the gamma intertrial coherence, so how consistent the phase is on gamma band, showed a very good sensitivity in predicting these individuals. Of course, like I said, this is also, the total sample size was 13, so it's a very small study, so very pilot finding, but however, quite exciting because, of course, like it would be significant if we could see 6 years before what will happen with TMS-EEG. So to wrap up this section on healthy and abnormal aging, so basically currently the field is showing that, or the literature is showing that healthy aging does not impact the TMS-EEG response morphology, so we still have the same peaks. However, there are some different differences in the amplitude, latency, and topography of these peaks. And of course, like the experimental paradigms modulate the peaks differently in young and older adults. And some of these alterations, like the P30, has shown promise in discriminating between healthy and demented subjects and evaluating the dementia severity. But like I said, it's still very contradictory. And one study found that Alzheimer's disease would be associated with an additional component. And this study was newly diagnosed individuals, so they did not have any medication. So. And also, like I said, there's a potential that TMS-EEG could predict who will convert to Alzheimer's disease and who will not. And the take-home message of this whole presentation, so TMS-EEG seems to be very safe across lifespan. So we did not see, none of the studies reported any significant safety concerns. And most research so far, as we of course know, has been focused on adults. And in children, TMS-EEG is still an underused method. Of course, for example, for ethical reasons, it's not so easy as in adults. And most of the studies so far have targeted the primary motor cortex and a few of the prefrontal areas, but all the other areas we haven't touched at all. So we don't know what happens in those other areas. We did not find any studies who looked at sex differences in relation to maturation and aging. Which of course could be relevant as many neuropsychiatric disorders have differences between the sexes. So we should first understand, of course, what happens in a healthy aging, and then start looking to understand why there's differences in the sexes in neuropsychiatric disorders. And most studies so far have been cross-sectional, so there's only one follow-up study, like a longitudinal study. But of course, to really understand how brain maturation and healthy aging and abnormal brain maturation and aging occur, we should have longitudinal follow-up studies. Okay, so here are my acknowledgements of this presentation. And I want to point out, so I am from a new lab. So this lab was established 3 months ago, and we are located in New York. So if you're ever in the neighborhood, come and say hello. And for those of you, if you are thinking about doing a postdoc in the US, keep our lab in mind. We have great resources and big plans for the future. And thank you so much for your attention, and happy to answer any questions you may have.
No, I cannot do it. Okay, sorry, questions. It's not working anymore. Okay.
Thank you very much. So regarding the large TP responses in children, I was wondering, so just for clarification, in the first study you showed the intensity was adjusted to rest motor threshold, which is larger in children, right? Yes. So the stimulation intensity was higher in children, which could in principle explain larger TP responses. But in the second study, it was not adjusted, you said?
Yeah, so in the superior frontal cortex, it was adjusted based on electric field. So we wanted to normalize so that in all the targets, in all the groups, had the same electric field. Okay, and you still saw larger responses in children, yes. Right? And based on the E-field estimates, did this result in larger like absolute stimulation intensities and maximum stimulator output? Like so if the E-field was matched, was still maybe due to the
difference in gyrification, in brain size, etc. So were the absolute stimulation intensities and percentage maximum stimulator output, were they different between children and?
Yeah. I mean, but
larger or?
A little bit, yeah. So I mean, this is like of course, like the endless problem in TMS-EEG field. Like, how do we normalize things? Like, should we normalize because of course, like the conventional way is the motor threshold, but what do you do in this kind of case when there's a substantial difference, like between the groups? So it impacts your results, but even if you do it with electric field, that's like so there's always like a problem.
So you have to choose your poison, right? I mean, yeah, of course. I'm just asking because I mean, higher intensities also mean louder stimulation, and that pertains to my second question. So many of these studies showed effects on the N100, which is also possibly confounded sometimes with an auditory TMS click sound potential, right? And often the topographies also look like typical frontocentral auditory components. So of all the studies you showed, also from different labs, how many of them did actually strongly control for possible auditory confounds, right? Because you could imagine that the auditory evoked potentials would also change with age, differ between different conditions of disease, etc.
Yeah, I mean, I'm very happy that you asked this, because this was, of course, the question, like the elephant in the room, so to speak. Because, of course, a lot of studies in adults have shown that a lot of the later response, like the N100, is confounded by artifacts. So yes, so of course this is a challenge. So currently, I don't think that we know, because of course the problem is that the findings are in adults, but we don't even know whether the N100 is the same in children and adults, because of course in children there's a prematuration process going on, whereas in adults it has already been finalized. So it could be that like auditory processing between these age groups are different, and that causes the larger N100 amplitude, or the N100 amplitude is just reflecting of very different processes. So this is, of course, because of course there hasn't been any also like pharmaceutical modulation in children or adolescents. So I guess like where's the next speaker? Isn't he doing a lot of pharmaceutical modulations? So we should need a study like this to kind of like understand what happens, and can we trust the N100 amplitude? Because it's kind of in all studies so far, it has been the promising biomarker, was promising in biomarker. So if it's just artifacts, of course, like we're back to zero.
Thank you.
So thank you very much for a nice and informative overview. I have two short methodological questions. When you are talking about children group, which age you mean exactly? Which age group of children from time window?
Oh, that's a very good question. So if I remember correctly, usually it's up to something like 13 years old.
13, it's already adolescents.
Yeah, I mean, so yeah, this is a very, very good question, because of course, like, for example, National Institute of Health has different criteria than, for example, what's seen in Europe. So yeah, very good question. I think so I can only speak for the studies that I was involved. Yeah. I think for those ones, the average age for children was 8.
Okay. Yeah.
But in some of these other studies, I remember seeing 14, something like that. Yeah. But it is totally like, we don't even know what we call children and adults, like we don't have agreement.
Okay. And second question, it was about this, what is minimal number of trials and a click of TMS is necessary to analyze this evoked potential? And I was wondering, because maybe different groups, different studies using different numbers of trials, and in itself single pulse TMS for a long time, somehow is a low frequency TMS, which is inhibitory, and then what is minimal number of trials is necessary to, let's say, analyze N100, because each click is strong enough, and how many we need?
Yes, I mean, so it is a very good question, and I don't think like if we would do a survey in this room, I would think that I will have so many answers, because I think like each lab has a different opinion and things like that. So I guess like we don't still really know what is the minimal number. So there is like a consensus, well, unconsensus paper coming where a lot of people from this room are also authors. So I think like we try to think about that, but I don't think that we reached any conclusions. I think like all authors gave a different opinion. So unfortunately, this is what it is.
Okay. Yeah. So then this means I'm not alone. Thank you. So around.
The range was like from 30 to 100 and something.
Yeah. I mean, so this paper is currently in the final steps of being published. But I mean, we collected opinions and tips from a lot of TMS-EEG researchers and tried to find a consensus. But like as you will see, there's still a lot to be done to find the consensus.
Thank you for the nice talk. I have a question about the data that you presented about M1 white matter. You presented a correlation between MD and AD and the amplitude, but the correlation was positive for both measures, but usually MD and AD are opposite measures of diffusion.
Oh, I see.
I don't know if you have any hypothesis of why, regardless of that, the correlation is positive for both measures.
Oh, that's an interesting question. I have to say that my knowledge of MRI is so limited, so I can't provide a good hypothesis. But yeah, in both of them we saw a strong correlations, and all the p-values were polyphenoly corrected. So it was a very strong. But that's a good point. We had an MRI expert in the study, but she, I guess, we didn't notice that point. Yeah, so I have to say I don't know.
Okay, thank you.
One question, and excuse my ignorance on that. In the correlation you established in children with white matter, gray matter and things, why you didn't look at the percentage of myelination, which is quite obvious to me. How much myelin these children had?
That's a good point. Well, at the time, we basically just wanted to see, is there a correlation? But like this study is still ongoing, because we are planning to also look at the connection on the prefrontal area. So maybe that's also something that we should look. But yeah, in this preliminary analysis, we did not look this.
Regarding the studies in Alzheimer's disease, you nicely presented the results on this component called P30. But this component is recorded in very different conditions across the studies.
Yes.
Starting from the target area. So in many studies is derived from M1, and then other studies from dorsolateral prefrontal cortex. So how do you think that it's kind of like useful to combine the conclusions from studies that start from very, very different target areas and networks?
Yeah. So this was just an overview, because of course we still have so few studies, so none of the results have really been replicated. It's just like a huge number of pilot studies. So you're absolutely right. We should have like look differently, different brain areas. I just pointed it out because the P30 had significant findings in all areas that have been done so far. So there could be something there. But of course, yeah, we need replications. Thank you.
Well, it's more a comment for you. I had the impression that, especially for developmental study, the number of subjects was very low, like 8 subjects, 7 subjects. So I'm wondering, how are those data, I mean, if those data are reliable in some way, because we know that there's a huge variability between subjects. So what's your impression after this review of those papers?
Well, of course, like the impression is that, well, everything seems to be very contradictory still, which could be based on the small sample sizes. So we need more studies and more replication to understand this. So you're absolutely right. A lot of these are very small sample sizes, which is a challenge. I think that the largest numbers are still like we're talking about less than 20 subjects, and that was like the largest studies. However, I would like to mention, because of course I come from US, so nowadays the biggest US funder, National Institute of Health, actually requires that all studies that you do have to be across lifespan, and if you don't include all results from across lifespan, you have to specify and justify, and they could have, so basically nowadays there's a huge push from National Institute of Health that we start, because they're not happy how researchers are just looking at very narrow age spans, but they're pushing more and more wider age spans. So hopefully we get, so this of course also TMS-EEG researchers have to start doing this from now on. So hopefully we get like a more extensive studies to confirm these findings.
Much more, we need more collaboration.
Yeah, more collaboration.
Any other question? No? Thank you.
Thank you.
Poster session and then last talk. Is that correct? Yeah. Sorry, just a moment. I need to...
Yeah, no worries.